It's our last talk of the day. Thanks for sticking through. And we're going to sort of end where we started. Tavi's talk took you into the brain to the level of neurons communicating with one another, electrically and chemically. And we've talked a lot about different and complementary ways to deal with addiction. And so we're going to end with the way which is trying to get at those chemical and electrical pathways in novel and very tantalizing ways. And I'll leave it at that and let you fill in the details.
Thank you very much. So I realize we're at the last talk and we are a team up here. So my name is Kathy Grant, and I'm the head of the Division of Neurosciences at the Primate Center here in Oregon. This is one of the largest national primate research centers. And I'm more of a behavioral pharmacologist, and Jen is more of a neurobiologist, and so that's why it's kind of a tag team here. So we'll just begin. So what I do as a behavioral pharmacologist and psychologist is I want to make sure that I have an animal model that informs the human situation. And so one of the best animal models or approaches for drug addiction is to see if the animal will voluntarily take the drug. Now, you can study actions of drug on the brain by just giving the drug, but if you want to study the addiction process, it's much more straightforward if the animal is choosing to take the drug. And so I spent several decades of my research doing this. And the non-human primate, in particular the macaque monkey, is a very informative animal model. And for all these reasons here. So we will show you some imaging data here and how we manipulate the brain. Also, in this model, oh, it's not showing here. Okay, so that's unfortunate. Let's go to the next slide. So in this model, we have to bring them in the lab. These animals are born here. They're reared with their mother until they are weaned. And that's about a 2-year-old animal. Adolescence in a macaque monkey is somewhere around 2.5 years old to about 4.5 years old. Then they enter into a young adult stage, 4.5, all the way up to about 6 years old. And then they go into their older adult stages. We bring them in, we do a way of getting them to experience intoxication voluntarily, and then we give them 22 hours a day where they can drink as much as they want, whenever they want. They always have water and they have meals. So this is the platform in which I also run a consortium of integrated neurosciences for the Alcohol Institute, and we compare the monkey data to the human data directly in this consortium. And so here's a human brain and here's a macaque brain. You can see that all the folding of the cortex is quite a bit different. It's a little bit more simplified in the macaque, but they have all the brain areas that humans have, and that's unlike some of the other animal models that is in neurobiological research. This slide is also pointing out one of the things we are doing, and that is to look at the stress axis, because there is a huge comorbidity between stress and addiction, and it's like a chicken-egg phenomenon. Particularly, like if you're using drugs heavily, that's very stressful, you're disrupting your life. So is that the source of stress? Well, you ask people, they say they're using the drugs because they're stressed. So are they using the drugs because they're stressed, or are they stressed because they're using the drugs? And our job is to disentangle this relationship, which has been a 15-year journey so far. Here is a very simplified model that we use in the consortium. And the amygdala, as you've heard, which is one of the emotional centers of the brain, is one of our main targets. And so I do want to point out that information comes into the brain through our sensory systems, our sight, our hearing, our feeling, touch. So we have direct contact from all our sensory information to our emotional centers. We also have a lot of talk with the prefrontal cortex and these emotional centers. And then the output is to our motor centers, so actually moving, taking action, and our stress and endocrine systems. And what we have found in our macaques is we can type our animals before they ever have any alcohol. One of the most important things about an animal model is that you can get information about the individual before they start using. And that's really hard to do in the human population. Good luck with the adolescents. That's hard too. We have laws that should prevent use, but we also know that 60% of our 8th graders have already drank alcohol. So. Anyway, the animals that are very aggressive have a different connectivity between, and this is without any drugs yet, between their amygdala, their emotional center, and their prefrontal cortex, which should be helping to give them decisions. And in this case, it happens to be prefrontal cortical area 9, and it's the right area from the right basolateral amygdala. That means it's on the same side of the brain. It's not that contralateral connection there. With information like this, then we can go in and we can see, will these aggressive animals that have this different connectivity, again measured in the same scanner that we use here for humans, will they go on to drink heavily or not? So that's one of the questions that we're asking there. I can give you a punchline on that, looking at the data beforehand. Aggression will predict how much alcohol these animals drink, but the aggressive acts do not get more aggressive with continued drinking. On the other hand, anxiety is not predictive of how much they're going to drink, but as they drink a lot, they become more and more anxious. So that's a very interesting finding for us. All right, we are now moving into the directly testing cognitive abilities in these monkeys. So one thing we've done is in their home cages, here's a macaque monkey in his home cage. They have touch screens now that they live with 24 hours a day. And I hope you can see it, it's a picture, it's a photograph of other monkeys. And what we found is that primates are very visually oriented. So just like we will play a video game, we won't get any money for it, we get points. We can go to different levels, but we're not getting juice, we're not getting drugs. We'll sit there and play this game, right? And it's the visual information. And what we found with these animals is that we don't have to reinforce them with food or with water or with juice in order to get them to play these games. All we have to do is change the visual information for them, and it's reinforcing enough. What that allows us to do is you don't have to superimpose all the brain pathways of ingestive behavior on top of the brain pathways for addiction. So it's one way of trying to separate out those pathways. The other thing is that the animals find some pictures very aversive, and some pictures they find that they prefer to look at, and we can use that information as well. Okay, so the one thing I wanted to say about this is the monkeys are engaging in a task called set shifting. This is one in which it's like playing a video game. You learn a task, and then the rules to that task change. And there's no outside information to tell you that. What you're doing that was correct is now wrong. You have to change your behavior, and then you can go to get it right. You can go to the next level, which is a more difficult task. And then as soon as you learn that, the rules change. And these animals, there are 8 levels to this particular set shift, and they fall out along a spectrum. So in a way, we can gauge how smart an animal is, and we can see if that will predict either protection or not for going on to drink heavily. The final thing, so that there was a better segue than that. And that is So what set shifting does is if you are not flexible in your cognitive behavior, you are not going to be able, when a rule changes, you're not going to be able to do that really well. Okay, so having learned the previous task is going to interfere with you being able to learn the next task. And so that's really what we wanted to understand. And there is areas of the brain, particularly the putamen, right here in the striatum. Everybody's been talking about the ventral striatum. That's the nucleus accumbens as a reward area. Virginia and I are very interested in the dorsal striatum. This is the motor output. This is the actual behavior that eventually comes out of the animal. Controlled here, the caudate is very well connected to the prefrontal cortex decision-making areas. The putamen is a bit more offline. It's more connected to the sensory input as well as the motor output, but not so much the associative cortex. Humans and monkeys, macaques in general, primates, are really good at multitasking. So that's one of the things that we humans are really good at. In fact, it sets us apart from probably the majority of the animal world. So we can be driving our car and thinking about our grocery list, no problem, right? Monkeys probably can do multitask as well. What we know is that the putamen can take care of habitual behavior, that behavior that's happening in the background. Sarah mentioned coffee and that people really like to have their coffee. I like to have my coffee with me. So I am really used to having a cup of coffee in my hand and being able to go through doors, go through meetings, do everything and not even really think, hold your coffee cup, right? It's just there. That's an example of multitasking where a motor behavior is happening, but you consciously don't have to think about it. Okay, so this is beyond just heartbeat and breathing. These are really kind of complex behaviors that just go on, and the putamen is the area of the brain that's necessary for that to happen. So now I'm going to switch this over to Jen, and she's going to tell us how we plan on manipulating.
Hi, so like Kathy said, we're interested in that other part of the striatum, the dorsal part. Can you hear me with this? Okay. And the reason why we're focusing on the putamen is because we think that the way that we view addiction is that it's an impairment in habits. So we're getting this upregulation of habit areas of the brain. And so we've done a lot of ex vivo work to show that this area of the striatum, the putamen, is highly upregulated with drugs of abuse, in particular addiction. And so that seemed to be a correlative effect. So, you know, if a monkey drinks a lot, we'll see that there's an increase in activity from this area. However, we don't know if it's actually causative. And so one way to be able to test that is to use DREADs. And so DREADs are mutated muscarinic receptors. They're G protein coupled receptors, but they no longer work in the normal way that they would in your brain. They no longer can be activated by acetylcholine, but they'll be activated by a drug called clozapine oxide that is not native to your body. So these receptors will only work in the presence of this drug that needs to be taken in either orally or through the bloodstream. And so the cool thing about these receptors is because they're not in your normal body, you can actually insert them wherever you decide you want to do. And so what we decided to do is to target these receptors to the putamen to see if we can actually activate this brain region when we wanted to. And so to do that, we did some stereotaxic surgeries within the non-human primate. This is actually a picture of a monkey within the MRI scanner. And so the stereotaxic injection is actually done within the scanner. The virus that is containing this, that will express this DREAD receptor, is also including a contrast agent. So you can actually see the signal here is where the virus is being put into the putamen. And so in real time, you can actually see the viral spread of this and make sure that you're actually getting it within the brain region that you're trying to target. So we know that we're getting localization. And so a lot of the work that we've done has been after the fact. So, you know, the monkey is injected with this virus, and then 30 days later, we kind of take the brain, we dissect it out, we look for the brain region where we put the virus in, and because it has a fluorescent tag, we know that these cells have taken up this receptor. We can do experiments where we can apply the CNO to see if if our receptors are actually working. But the cool thing now is that we've found a way to use fMRI to see that these receptors are functioning, and then they're in the right place within the live monkey. So, for example, in this image here, the monkey was injected with a dread that when CNO is on board, it'll actually shut down neural activity. And so we placed that specifically into the putamen. So if we look at the putamen, this is the bold signal here. And this bar here is where CNO is being administered IV. And we find that with CNO on board, we get this decrease in the bold signal, saying that that the CNO is activating the dread receptors that are in the putamen, decreasing it. But if we look in the cortical regions that's upstream of the putamen, so this would be the somatosensory cortex, we find that this is not affected. And so we can specifically inhibit or excite specific brain regions depending on where we so I think this is just a new technique that we're trying to impart to say, are these brain regions important for these behavioral effects that we're looking at?
Okay, so now I'm going to jump back in and tell you what we predict. So by taking the putamen offline, so again, this is a receptor that we put into the brain. It's normally not there. There's nothing normally in the brain that's going to activate this receptor. So it's only when we inject the drug that this receptor becomes active. And by taking it offline like this, we predict that if an animal is very habitual, we will shift that behavior over into a more prefrontally mediated behavior, right? So that's our prediction. There's several amazing things about, and what I'm talking about for the people on the other side of the room is the far right-hand lower graph. There are several amazing things about this graph, mostly because it's in vivo. So what we can do that we would never be able to do before is know that when she says virus, that's just the thing that allows us to target and get into the cell. Viruses get into the cell. It has a cargo, a DNA cargo load. That's the receptor that's being delivered, right? Well, when you deliver it, you don't know if it's active or not. And before, we always had to take the brain to see, retrospectively, was it active? Now, prospectively, we know it's in there. We know it's active. We know how much drug. We also know from brain to brain, individual differences will occur. Brains will take up different amounts of this virus. So before we ever start our experiment, we can know how much CNO we need to deliver to each animal before we do the experiment. So we can equalize across animals, and we can get a better idea of when we shut off the putamen, do we actually make behavior more flexible? Are they more cognitively flexible? Are we forcing the animal to use its prefrontal cortex a little bit more? That's the main thing. And the other thing on the other side of the graph is to show, again, with functional connectivity, this was how much alcohol the animal eventually drank, but had not drank yet when we took this measure right here. So it's a way of predicting, before the animal ever started to drink, that connectivity between the amygdala and the prefrontal cortex is predicting who's going to be a heavy drinker and who's not going to be a heavy drinker. I'm running out of time, so I'm just going to go straight to a study that I really hope you can appreciate, and that is repeated abstinence. So this was inspired by a court, by a document, a study that was done with human alcoholics that were court referred to an abstinence program. So it wasn't voluntary, and that's the important part. So a group of alcoholics had to go in, and they were compared to alcoholics that voluntarily sought treatment. And they had 28 days of abstinence, and they wanted to see which group did better. And in fact, the imposed abstinence group did far worse than the voluntary abstinence group, right? And so our monkeys really would have no idea after 12 months of drinking, and anytime they wanted, 22 hours a day, we took the alcohol away from them, right? So they didn't voluntarily do that, and they had no idea why that happened. So that's a little bit more removed from the human situation, but just imposed abstinence on these animals. And this is, over here, how much the animals drank in their 12-month average. Each animal is a dot, just 1 month pre-abstinence. And then abstinence for 28 days was imposed. And everybody early drank more in abstinence. This was a 3-month period, so the first month versus the second month. We then impose abstinence again, and everyone's drinking, even the lowest drinking animals all increase drinking. So imposed abstinence is something that truly increases drinking. This is the blood ethanol concentrations. This is 80 milligram percent here, or 0.08. And even the very low drinkers after the second abstinence was drinking up into a very intoxicated level. I only have one more slide, and that is to emphasize that this effect was mainly due to the heaviest drinkers. So the lighter drinkers imposed abstinence did not really change their intake much. It was the heaviest drinkers that fared the worst with the imposed abstinence. And then just because the judge did such a good job, I do want to say that here's a brain, here's an alcoholic brain. Here's a person who's not an alcoholic. Lifetime consumption of alcohol was 40 kilograms. The alcoholic, 1,500 kilograms in his lifetime. Drank for about 30 years, 33 years, but had been abstinent for over a year. And you can still see the brain is still in recovery here. And we don't know if the brain will ever recover. Animal models will help us with that. But again, working with people who would have this kind of damage is going to be very different. The expectations will have to be different than working with a fully functioning brain. And this is a summary of everything I just said, and it's a few minutes after 3, so I'll end there, and thank you for your attention.